The first year of the coronavirus pandemic was challenging, stressful, and very scary. If you were reading the news around that time, when the vaccines appeared and started to offer all of us a glimpse of hope, you saw headlines like this: how could anything developed this quickly be safe? And no matter what news site you were looking at, the question was the same. So people understood that the RNA vaccines developed by Pfizer and Moderna were based on a completely different mechanism and different constituents than all the vaccines we received as children. And they were developed against a virus that didn't even exist a year before. So when I was talking to my friends around this time, friends who know that I'm working on RNA biology, they were asking me two questions. They were asking me how the vaccines work, and they were asking me if I would consider them safe, considering how quickly they were developed. And I'm going to answer those two questions, starting with the second one. So is it possible to develop a safe vaccine based on a brand new mechanism from scratch in the matter of months? Probably not, but luckily that's not what happened. The vaccines we all received have decades of basic research behind them. And I wanted to start with showing you a page from the laboratory notebook of Robert Maloney from 1989, so over 30 years ago, where he describes an experiment which is the very basic of the RNA vaccines. So in this experiment, Maloney took an RNA molecule and encapsulated it in a lipid nanoparticle. And I don't need you to understand these concepts yet. I'm going to explain it, but you can just appreciate it visually. He injected this complex in mice, which led to the production of a protein. Now, a next important player in this field is Katalin Karikó. And I'm not only showing you her because she's Hungarian just like me, and I'm very proud to hopefully have a new Hungarian Nobel Prize winner in the next few years. I'm showing you her because her work is, she was working on RNA modifications for decades. And she didn't know that it will lead to the vaccine. And what she is doing is actually very relevant for my own work, and our lab even occasionally collaborates with her. But mostly I wanted to show you her because I wanted to mention an article which is partially her work, but mainly based on her work from 2018, so the year before coronavirus even existed. And it has this very complicated title: Nucleoside-Modified mRNA Immunization Elicits Influenza Virus Hemagglutinin Stalk-Specific Antibodies. Now, what this actually means is that they had an RNA vaccine against influenza virus the year before corona existed. And this RNA vaccine never really got to production for humans because pharmacological companies are profit-based companies, and at the time it wasn't profitable, but the technology was there. So with that, I want to move to the second question about how the vaccine works. So coronavirus has on its surface a so-called spike protein. This is, again, something you probably heard if you were reading the news in corona time. It's a protein which is necessary for the immunogenicity of the virus, and it's one of the first things that your immune system meets when you get infected. So the vaccine consists of a lipid nanoparticle that encapsulates an RNA molecule. The lipid nanoparticle, you can imagine it as a drop of oil. And the RNA molecule inside codes for the spike protein, not the entire virus, just this one protein on its surface. What coding for the spike protein means is that when this RNA molecule gets into your cells after you get the vaccine, your cellular machinery can read the code contained in this RNA molecule, and based on that code, it can create this spike protein. So after this, the spike protein gets transported to the surface of the cell and presented to your immune system, meaning that your immune system will learn to recognize this spike protein. And when you get infected by the virus, it's not going to be something new. It's going to be something that your immune system already learned to recognize and can react against much faster, not giving time to the virus to proliferate and leading to a much milder infection or none at all. Now, I keep saying that the main constituent of the vaccine is RNA, but what is actually RNA? So the most accepted hypothesis of how life developed on Earth is that it all started with a time where everything was made up of RNA. So all the functions that in today's life are performed by RNA, DNA, proteins, and lipids were only performed by RNA. which really means that RNA is a molecule that does absolutely everything. It carries information between different parts of the cell. It can itself encode protein, but it is also part of the machinery that makes proteins. It can work as an enzyme and enhance the speed of chemical reactions in your body, and it can even be a heritable genetic material. And it can look like this, or this, or this, depending on its function and the artist who drew it. Now, if you look at school books, they will tell you that RNA consists of 4 basic building blocks that we mark with the characters A, C, G, and U. And you can imagine it as putting pieces of Lego one after another. Now, I told you that RNA does everything, and you might assume that doing everything needs a more complicated regulation than what a simple code base of 4 characters can do. So in reality, RNA looks a lot more like this, where every one of these 4 basic building blocks can have hundreds of different chemical marks, tiny modifications on them that regulate literally everything about their function. So for example, these modifications will regulate the lifespan of RNA. So is a certain RNA molecule going to spend minutes in your cell and get degraded, or is it going to be there even for an entire week? It regulates the localization of the RNA. So is it going to be in a certain part of the cell or a different part, or can it maybe move in between those parts? And it regulates the amount. Are you going to have just a few copies of it, or are you going to have tens of thousands? And in the case of the vaccine, the modifications do two important things. First of all, they increase the stability. So you can store it at minus 20 degrees instead of minus 80, and it's not going to right away degrade when you thaw a vial. And it also helps this RNA molecule to avoid your immune system, so it's not going to be attacked by your immune recognition, and it can elicit its function and code for the spike protein. But in the entire human physiology, regulating all these functions means that RNA modifications affect cancer progression, regulate immune response to infections, regulate human embryonic development, affect your metabolism and aging, making it literally the most interesting thing in the world. Now, in my research, I normally start with doing laboratory work, meaning taking RNA and putting together some chemicals, pipetting, centrifuging, and all this will lead to a small amount of transparent liquid, which then I can load on different very expensive machines that we have in the lab, which will analyze the content of this transparent liquid and give us some really, really awesome data, which I follow up with computational analysis. And I do all this in order to develop new methods, to find new RNA modifications, or to have better detection methods for the ones we already know. I use molecular biology techniques to understand the basic mechanism of RNA modifications. I try to develop tools for biotechnology. I want to understand the relevance and context of RNA modifications in different diseases, and I develop computational pipelines. Now, strictly speaking, my research has nothing to do with vaccines. And for most of the time, Katalin Karikó's research had nothing to do with vaccines either. So one of the main challenges of doing basic research is that we can never know if what we are doing is going to have any practical benefits, and if yes, what is it going to be? But luckily for us, the most unique thing that makes us human is that we are able to appreciate learning, knowledge, new pieces of information, even just for their own beauty. With that, I want to thank you for your attention, and I'm happy to answer questions.
Thank you, Anna.
Hi, Michal from the Weizmann Institute. Thanks for your talk. I was just curious, you talked about, in the last slide, you talked about your research. Could you give us an example of a specific tool or method that you developed in a specific context? I would really like to hear that.
Okay. So maybe one example would be that I planned we call it a massively parallel reporter assay, which basically means that you computationally design a lot of different RNA or DNA molecules. So I told you that it's basically like putting pieces of Lego together. So I can decide how to put the pieces one after another, and I can decide it thousands of times. And when we try to find RNA modifications in your own body, your own body is a mess, so it's really hard to do. But if I can design where exactly I want to look, it's much easier to find rules. So maybe that's an example that I keep using in my research.
Hi, thanks for the talk. Roy from the Hebrew University. In order to conduct such a multidisciplinary research, you must have a broad education in biology and computer science. Maybe you can tell us a bit about how did you get and what do you enjoy more, the biology or the computational aspects of your work?
Okay. So my undergraduate degree is in molecular biotechnology, which is kind of a mixture of chemistry, physics, and biology. And I did a bunch of internships in different molecular biology parts. I was doing some research in cancer and influenza virus and macrophages in cardiac disease and all kinds of things. So I learned a lot of techniques of molecular biology. I never learned programming. I still don't know programming, but I can copy-paste. And so I kind of enjoy both, and I'm doing half-half. The computational part is definitely more challenging because I'm kind of trying to figure it out myself, but with a lot of help from my lab members who are much smarter than me.
Hi, I'm Dana from the Hebrew University. Thank you, Anna. Great talk. I know you said you weren't specifically focused on vaccines, but nevertheless, can you give a little bit of insight on why RNA vaccines were so much faster to develop than traditional, and whether we can anticipate them replacing in the future traditional vaccines?
Okay. So it's really easy to develop, basically very much connected to the answer I gave to the first question. So we can just take a sequence of RNA, design it as we want. We can design it to code for the spike protein of coronavirus, but we can design it to code for anything else. And the mechanism exists to synthesize it. It's a matter of days. So basically, a brand new production pipeline for a new vaccine takes a few weeks. So I think Pfizer was saying that it's about 6 weeks to try to adjust their production to work against a different variety of the same virus or a different virus. And it's definitely, I think, a very promising method for the future. And it's a bit hard for me to tell if it's going to be widespread or how widespread it's going to be because of what I mentioned about profitability. So if there's a vaccine against influenza that pharma companies are producing and you have to get it every year, it's not going to be profitable for them to buy the machines to make something new that you might even not have to get it every year because you are paying for them, and it's like a lot of political and financial questions that contribute to this.
Yeah, I'd like to add up on that. I participated last summer in a conference on RNA therapeutics in Arizona, where 1,000 people were coming with sparkling excitement. Today, after there was no drug based on RNA that existed, there are 18 different therapeutics based on RNA. And it is much faster, much cheaper, and much easier. All it took is a pandemic, because the FDA had to approve a human test. Moderna existed for 20 years. Nobody gave them the permit to push a syringe into a human arm until there was no other option. And the people at the conference were totally excited. It was really a very inspiring experience. So I definitely believe that it would make a very big difference. Probably by next year, the influenza vaccine will be another RNA one. This being said, there are people who say, this is too much, we are getting too close to the genome, we are over-immunized already. There will be social discussion. Perhaps that's good too, I don't know. And that was a great talk. Thank you.
Thank you.